Hello and welcome to Navigating Autoimmunity through Personalized Microbial Insights. My name is Adair Anderson. I am the product marketing educator here at Vibrant and want to go over some quick housekeeping items before we get started. All attendees are in listen-only mode. If you have questions during the presentation, please submit them as a comment. There will be time at the end for Q&A. We'll go through all your comments at that time to make sure we address them. And now I am pleased to introduce our speaker, Dr. Sarah J. Zielsdorf. Dr. Zielsdorf is an Institute of Functional Medicine certified practitioner and is board certified in internal medicine. As the owner and medical director of Motivated Medicine, an innovative consultative medicine practice in West Chicago, Illinois, she is a skilled diagnostician, microbiologist, and passionate teacher. In addition, Dr. Z serves as a medical and research advisor and the educational director at the Low Dose Naltrexone Research Trust. She created the 2020 through 2024 LDN guides and is an author featured in the LDN book, Volume 2 and 3, and New Horizons. The motivated medicine approach that Dr. Z developed and continues to evolve is rooted in translational medicine, which bridges the gap between current clinical research worldwide and the direct care received from the physician. Dr. Z's open-minded approach to treatment is informed by data, advanced diagnostic testing, whole body wellness, tried and true conventional medicine, and so much more. Take it away, Dr. Z.
Hi, everyone. Thank you for joining me on this Tuesday afternoon. I'm going to go ahead and go into my presentation now. Again, thank you, Adair, and thank you, Vibrant, for having me. I am going to go through this presentation, and please go ahead and ask your questions. As she said, we will try and answer them at the end. So yes, that is about me. I did want to highlight the fact that it's very apropos that I did this training and my background is in infectious disease and microbiology, but I, in fact, am an autoimmune disease patient and I have Hashimoto's. And I'm going to use that as an example. And so, yes, this is my practice. I really deal with medical mysteries. And so things, I'm sure you feel this way too, things are not getting easier. They're just getting more and more complicated. And so there's always that adage, when in doubt, start with the gut. And maybe that's a little simplified, but it's not far off. And so I'm in the western suburbs of Chicago, and I treat a lot of Midwestern patients, but I see patients from around the country. I have no conflicts of interest to disclose. These are our learning objectives, which you can read here. And so I'm going to hopefully review some relationships between specific microbial findings that you will be able to utilize come next day, and that I really want to have you start to look at and to recognize, and how using Vibrant testing can really help to tailor treatments. So I don't know if you feel this way, but I really believe that we are at an inflection point in medicine, and we really have two versions of medicine. We have allopathic medicine that is dictated by insurance companies, and we get a certain finite time to put in an ICD-10 code and have a certain set number of limited labs that are covered, and we're having to make treatment decisions in 5 minutes or less. And then there is the really health-focused translational medicine that is taking everything that we know from the bench to the bedside in real time. And as we know, we really are slow to adapt to change. Things that are coming out, new insights, new wisdom, the average lag between what's coming out in the journal articles and basic research to us changing medicine is 17 years or more. And so really how I practice is N equals 1 medicine. Every single patient is unique. But we're limited by current lab data. So where do we focus? How do we really do personalized and precision testing? And how do we guide our treatment decisions? And I will discuss the way that I go about this. But I want to really focus on autoimmune disease today because it is everywhere. And I will argue that we all have autoimmunity, but we don't all have end organ damage constituting a specific autoimmune disease, which is often systemic. Um, but bottom line is the prevalence of autoimmunity and autoimmune diseases are increasing. And this is recognized in immunology and regular allopathic medicine. At least it's being given lip service. I don't think it's lip service. I do think that the world is looking and the scientists are voicing and saying we are seeing a lot, and there is a lot that is changing. What is not changing are our genes. We humans are not evolving that fast. So, what has changed for us to become so autoreactive? And that really is because of multifactorial changes in our environment, our food, our constant contact with xenobiotics. So, especially estrogen disruptors, air pollution, infections, chronic infections, viral infections, stress, um, other environmental changes. And so, really, it's a huge burden to society. Those that have autoimmune disease often do not get diagnosed for up to 10 years. They go through many different doctors. They have high costs. They have a lot of suffering, and there is poor diagnostics, poor treatment options, and it is not based on prevention. We know that once you develop one autoimmune disease, you're likely to develop more. And I want to use Hashimoto's as an example. And so I'm a really passionate student of history. And this is the disease that I have that really took my youth. I started to get sick in my teens. I was finally diagnosed as hypothyroid in my going into my third year of undergraduate. And I remained sick. But let's discuss, if you've never heard about Hashimoto's, let's talk about Hashimoto. Was named as an eponym of Hikaru Hashimoto, and he lived here from 1881 to 1934. And he first described from histology samples from 4 thyroids of women that were all surgically excised. They all had autoimmune thyroiditis, but he described this as struma lymphadomatosa. So really a lymphomatous goiter. And so a large thyroid that had tissue within it. The soft tissue was infiltrated by lymph cells or white blood cells. And so he published his findings in the gold standard journal, which was German, the Archiv für klinische Chirurgie, and that was 1912. So let's talk about this right now. Hashimoto's is not simply having hypothyroidism. It does not mean that you just have hypothyroidism. Hashimoto's is the state of inflammation, and that can be defined as the presence of having thyroid peroxidase or thyroglobulin antibodies. But do not be confused, or do not be mistaken that you don't have to have antibodies. You can be seronegative. And really that indicates immune system burnout or burned out Hashis. A patient can have over 300 symptoms of Hashimoto's, and that includes all of the typical hypothyroid symptoms, but you can also have hyper symptoms in a thyrotoxic state. And so patients can swing hyper and hypothyroid, but in the early phase, you can have weight loss if there is a hyper phase. As symptoms progress and as the disease progresses, you can get hypothyroidism and thus all of these symptoms and more. And I often refer to Hashimoto's as the canary in the coal mine because we really do believe that this is a gateway autoimmune disease. And for many, it's a first autoimmune disease that's diagnosed. Patients can collect them, including celiac. About 10% of Hashimoto's patients have concomitant frank celiac disease. More than 90% will be sensitive to gluten. And I will argue that we really should maintain that those that have Hashimoto's be strictly gluten-free and maintained with celiac precautions. And I'll talk about that. But also rheumatoid arthritis, type 1 diabetes, Sjogren's, vitiligo. These are very common crossovers that I see. Patients can sometimes really get early diagnosis, often found because their patients, the patient is advocating it's often found by themselves, or they're advocating for family members because they see strong family histories of autoimmune disease or thyroid, which tends to run in women. And again, in early cases, if you use intense lifestyle modifications and adjunctive therapies, you can improve quality of life and sometimes prevent and get this to be in remission and prevent the destruction of the thyroid gland. And so, what is Hashimoto's thyroiditis? That is inflammation of the soft tissue or the parenchyma of the gland. It's chronic inflammation of the thyroid. There is risk of autoimmune mediated destruction of the gland at about 5% risk per year progression to overt hypothyroidism. So we can see that there's a long latent period, of course, in a lot of cases, which lasts up to a decade. Now, the most common cause for someone to have rapid destruction of the gland is going to be due to a virus or due to pregnancy-induced thyroiditis. And there is a risk of future malignancy, most commonly primary lymphoma of the thyroid. So this is really important. And we really want to diagnose this with an ultrasound. Remember, I said that you can have Hashimoto's without having thyroid antibodies. And this is a clinical spectrum. And we see, really, really appreciate this, this chart, this Venn diagram that shows that there is quite a bit of crossover in what we call AITD, or autoimmune thyroid disease. And this is a spectrum everywhere between straight Hashimoto's, which tends to be either TPO or TG antibody, to a crossover with Graves, where patients can have both. Patients are straight Graves, or patients have Graves or with atrophic thyroiditis. And so, taking this from a paper, they say, due to a complex and still poorly understood process. And I'm going to argue, how autoimmune disease starts. And so this is very, very frustrating. They say, well, we don't know why this happens, but we get an autoimmune mediated infiltration of the thyroid gland by T cells and B cells of the immune system. And this is inflammatory. And so what I will talk about is that, again, the definition is a lymphocytic infiltration of the thyroid gland. That's a histologic diagnosis. So I would argue that autoimmune disease is a lymphocytic infiltration of an end organ, a gland, or tissue. So patients collect autoimmune diseases. So if this is the same underlying pathophysiology, why are there no autoimmunologists, right? And so that's kind of the joke. I say that we in this field really are autoimmunologists. And so autoimmunity to autoimmune disease expression is a continuum. And it's one that I take as a three-legged stool. And so when we look at this, we have three conditions that are required. And how I teach my patients, I say, one, there has to be a genetic predisposition. Well, if you're a white woman of European descent, well, there you go. Every single one has a genetic predisposition, whether it's in their family or not. Two, an environmental trigger or triggers, which we all have. And three, intestinal permeability, or the so-called leaky gut. Now, please do not say that in front of allopathic doctors who don't believe in that. But I do this every year as a exercise. An exercise, I pull the PubMed list and I type in search intestinal permeability, and we can see so far in 2024, there aren't as many, but we can see every year it has gone up, this so-called leaky gut. There are almost 26,000 results of a disease or a state that, quote, does not exist. Well, it does exist. And I'm going to review the aspects of that. And this is a wonderful review paper if you need to share with patients about the human intestinal barrier function. And there are many, many guidelines on this now. And I'm going to talk about that. And I want to just walk through the human intestinal barrier just as a review. And this is a schematic of not all, but a lot of the major affecting factors for this. And so there are several layers providing protection against microbial invasion. And if we think about it, this gut lining sees more antigens every single day than something like the skin would see in a person's lifetime. And yes, we get cuts in the skin and things like that, and we have to fight off pathogens, but we don't get the onslaught that we get by swallowing our saliva all day or every time we eat or drink something. And so when we think about this, I'm going to start from the inner part to the outer part, or depending on how you look at this, the lumen is going to be the food side on the top, if we start at the top of the diagram. But the intestinal lumen contains AMPs, or antimicrobial peptides, and secreted immunoglobulins, or SIGA, secretory IgA, which I'm going to talk about, and commensal bacteria, which are neither good nor bad. They are important placeholders. So basically, they are that shag carpet, which inhibits that colonization and overgrowth of pathogens by competitive inhibition and by production of barrier protective butyrate and other fermented materials. A mucus layer covers the intestinal surface. It provides a physical barrier. And this single cell layer, which is the epithelial layer, it is one single layer of epithelial cells. And it's sealed by tight junctions, which includes occludin, claudin, and zonulin 1, which prevents the passage of macromolecules, large molecules, paracellularly, or where they should not go through the cell. And this layer also has other cells, intraepithelial lymphocytes, M cells, which are really important, big ticket cells which overlie the mucosal immune system, like the Peyer's patches and other lymphoid follicles. We have mucus-producing goblet cells, which I'll point out in the next slide, and Paneth cells, which produce bactericidin. And then finally, we have the lamina propria, which is containing a large amount of immune cells. So that's the immune system that is peeking through the walls to see what's on the luminal side. And we can see that the innate immune system is going to be represented in the lamina propria. So that's the macrophages, the dendritic cells, and the mast cells, and the adaptive immune system, so T cells and IgA producing plasma cells. And so there are also cells of the central and in the enteric nervous system, which innervate in the lamina propria. So remember, we've got the important connection between the gut and the brain, literally in these connections. And so there are many, many factors which affect the health of the intestinal barrier and the function of it, including whether you've got pathogenic bacteria, and the example here is going to be enteropathogenic E. coli or EPEC, a high-fat diet, and I talk about improper fats, seed oils, lipopolysaccharide or LPS, which is a sugar and gram-negative bacteria, which I'll talk about as endotoxin. Drugs such as NSAIDs and PPIs, which degrade the lining of the slime layer, that mucus layer, and then if you've got food allergens or gliadin proteins, so from gluten, which is highly reactive for some people via molecular mimicry. So next. So again, these are the three major components of the intestinal mucosal barrier, which I really want you to remember. We've got the lumen, and we've got the epithelium with its mucus layer, and the lamina propria. And I'm just highlighting this mucosal system with mucin and goblet cells and the production of antimicrobial peptides. And we can see here that via eating good dietary fibers, we're producing lots of nutrients in the form of short-chain fatty acids. Butyrate is the most well-known. And we can see we have both pro and anti-inflammatory signals in this state. So I want to talk about our knowledge of the microbiome. And I want to just, as a microbiologist, I will say it's woefully small, our knowledge. And I often think of it as outer space or the ocean. This picture is of the Mariana Trench, talking about how deep it is and how it's kind of a mystery, down in the depths. It's difficult to get to, it's difficult to do experiments, it's difficult to do research. And I think of the microbiome that way. Almost everything that we know, the preprints that really developed out of COVID, starting around right before around 2018, 2019, we started to have technology that really started to explode. And we're going to talk about these keystone species and talking about how, doctors and other researchers will talk about how much we know, but we just started learning about it. I'm going to talk about Akkermansia, other core species. This bug wasn't even elucidated, when I was in grad school, and this is not that long ago. So when I talk about the microbiome, I just really want to kind of talk about what I'm using it as a generic term, like butter, but not a brand of butter. The microbiome is classically composed of bacteria and viruses and phages and yeasts and parasites and all sorts of things. So when we're talking about very, very specific granular level of just the yeast in a person's microbiome, that would be their mycobiome, or just their viral components, and that's their virome or phagiome. So and then we have to talk about the interaction between the microbiome and the immune system and the endocrine system. So for instance, there is an estrobolome, which is really, really important. And so we have these multidimensional host and microorganism interactions. And so remember, we have tenfold more phages, which are phages eat bacteria. They're tenfold more than bacteria, and these are new, and we don't know much about them, and they play roles in these interactions. So this is a really nice paper, and I included these translational medicine papers and some good data, some good articles that are starting to just talk about the idea of dysbiosis and imbalances in the microbiome and what is considered normal flora, and that there is a bidirectional interplay between the gut microbiome and all of these organs. And so what's so interesting is this is a really nice diagram that is just introducing the idea of not only a gut-brain axis, but a gut-lung axis and a gut-skin axis, and talking about how the dysbiosis that occurs in the human microbiome is going to be implicated for not only right functioning of organs, but if there's disruption to the microbial homeostasis, you can get malfunctioning in these affected organs and the progression of many, many related diseases. And so what I'm arguing is that you are treating the gut, but you are looking forward toward many aspects of a patient's health or disease state. So next, I could not go forward without, again, discussing and reviewing secretory IgA. So secretory IgA, these are what we call the first responders of the immune system. And I want to really remind people that you should be measuring SIgA, and that is represented on the Vibrant test. It is the most abundantly produced antibody in humans. And the cell that secretes IgA these are plasma cells, and they're in mucosal tissues, mostly in the gut, and this can help neutralize toxins and infections as well. And so this is a nice paper, nice review on that. And again, secretory IgA is going to have multifunctional interactions. There are many interactions between this immunoglobulin and both pathogenic and non-pathogenic bacteria in the intestinal mucosa. So we can see in this slide that again, in A and B, you are getting the prevention of pro-inflammatory responses and the preservation of the epithelial barrier. And so secretory IgA is neutralizing toxins, and you are stopping the enemy at the gates. Versus if it can't, then you're going to have higher level innate and then adaptive immune system that is going to get triggered. Now, the other thing that is really, really important for secretory IgA is that you are going to select and maintain the non-pathogenic bacteria or the commensal microbiome. These are the guys that, again, make up a slime layer and develop a biofilm, meaning a good biofilm that is enriching the mucus rich environment and supporting a healthy intestinal epithelial barrier. So I want to focus on changing the paradigm. I want to shift this. Back in 2008, 2009, 2012, what did we do for those with autoimmune disease? We restricted their diet. We put patients on very, very strict elimination diets, including things like autoimmune paleo. And we caused a lot of harm. And in susceptible patients, mainly women, we had physical and psychological consequences, patients who lost their oral tolerance, patients that had developed worsening eating disorders where they already had dysfunctional relationships with food. And so we don't want to restrict the diet first, with the exception of things like the most inflammatory foods like gluten and dairy. We want to test. We do not want to guess. And so the red flags are going to be where you are recognizing for patients that they have low secretory IgA. It is much easier to help a patient with high secretory IgA, meaning that there are tons of soldiers swarming the gates and fighting off stuff rather than no one to defend, and a very burned out immune system in the gut with poor amounts of first responders. Of course, if there are pathogens, you want to treat them. If there's low microbial diversity and thus low short chain fatty acid production, depletion of their keystone species and poor microbial diversity. So if they've been on chronic antibiotics, including for Lyme, failure to recognize pathogenic biofilms and failure to treat underlying endocrinopathies. So if there are hormonal issues, which many patients with autoimmune are going to have, and failure to address the underlying trauma. That's incredibly important. And so that also goes to bear, brings to bear the importance of understanding that vagus nerve. And it's not just toning the vagus nerve. Some patients actually do worse if you do that. And it's really about understanding and balancing that, that connection between the gut and the brain. So a reminder, we have tons of normal flora. And this is another great paper on what is considered core keystone species in different areas of the body. And I'm going to focus on the gut, but this is a nice review. So what are keystone species? And I want to argue, so this was a nice review as of 2021, but I will tell you that this is an ongoing process. We really are in this next generation of research to identify these species and look at host gut microbiome interplay. However, I will say that we're still limited by technology. This is a really nice just review of how are they determining what are keystone species in the gut. And they are doing things like shotgun sequencing and very large metagenomic analysis of the microbiome to look at signatures for the abundance of what is in high concentration in healthy individuals, and thus comparing them, looking at the power via the number of samples and the depth of analysis to understand what are abundant keystone species in healthy individuals versus what is lacked by those that are not healthy. And we can see also that plays into how short chain fatty acids are or are not produced, thus influencing further gut microbial diversity. So this is just a nice review, and this is not meant to be all inclusive, but I have highlighted, we've got a storm going on here, so you can probably hear that in the background. I'm highlighting just a few, and mainly the things that I want to talk about are two here. We've got Faecalibacterium prausnitii and Ruminococcus bromii, and these are butyrate producers. Again, the more butyrate we can naturally make, and these are really nice anchoring probiotic species, the more we have naturally, and I see these that are depleted, the more we have, the more they are producing butyrate, which thus feeds the good guys and then produces more good probiotic species. When these are lost, it is really tough to establish these keystone species. Obviously, bifidolongum is one of the OG species that we talk about. It's really important for the health of babies' guts. We talk about the fact that it helps to degrade HMOs, or human milk oligosaccharides, and really helps to degrade complex carbs. And then I'm going to talk a lot about Akkermansia, which helps to degrade the mucin layer. And so, yes, Akkermansia, mucinophilia. I talk about it as the Goldilocks bacterium. Too much is toxic, too little is bad. It is a mucin degrading bacterium, and it is inversely associated with diabetes and chronic inflammation. It promotes microbial diversity, and a pearl is we want to use supplements if we have to, but mainly getting patients to eat their dark berries. Sometimes we use functional foods that have freeze-dried amounts that they can make shakes or gut healing shakes, things like that, to fuel Akkermansia growth. Now, I will say if a patient does not have any Akkermansia as a starter, it can be really tricky to enrich these Akkermansia levels. And this is a nice paper talking about how is there a direct concept between the presence of Akkermansia and disease? And we can see that in both obesity, diabetes, type 2 diabetes, and also in those patients that have had gastric bypasses. We can see that there is a relationship, meaning that the lower that Akkermansia is, the more problematic that is for disease. And if you are treated, the obesity is treated, the type 2 diabetes is treated, that you're going to have a changed microbiome. So I want to talk a little bit about biofilms. This reminds me of a Dr. Seuss book. Let's have a little talk about Tweedle beetles. But I want to talk a little bit about biofilms, which is a really big topic. And there's a lot that I see in the kind of wellness sphere of people just talking about biofilms, but not really understanding the huge amount of data and science of what a biofilm is at the microbial level. And so I want to break this down. This is a really nice review paper, and I'm just going to discuss it a little bit. And so this figure is of an intestinal immune barrier. And so in homeostasis, on the green side, bacteria will sense the density of themselves or the surrounding bacteria, and they secrete these AIs, which are autoinducers to the extracellular environment. And when signal molecules reach a certain threshold, we have something called quorum sensing, or QS. And quorum sensing was first really identified in the early 2000s. My microbiology professor, shout out to Dr. Ann Morris Hook, said that this was going to be one of the most important things that was understood, and she was right. So basically, you get this hive mindset where if the threshold of bacterial colonies can get to a certain level of favorability, you will turn from off to on, which is then recognized, and then you get target gene expression, which controls something called EPS, or bacterial extracellular polysaccharides, which are these sugars, which are both being synthesized, they're causing aggregation, colonization, this slime layer of biofilm to be formed. And so that's how these colonies enrich and are protected. And at this green level, this homeostasis status, you have mucus, antimicrobial peptides, and intestinally secreted by B cells secretory, or SIGA. And then you get also production of how to isolate intestinal bacteria. You also have anti-inflammatory cytokines like IL-4, IL-5, IL-10 to protect the mucosa epithelium, and you fix the good guys, the symbiotic flora and the protective mucus. You have dendritic cells, which produce a tolerant response, which downregulates that pro-inflammatory kind of juggernaut, that NF-kappa B, that signaling cascade. And then this symbiotic relationship, everything is beautiful. You have the symphony between organism and symbiotic flora. on the red side, for the inflammatory or inflammation status, you get a loss of the intestinal barrier or that leakiness. You get translocation of the bacteria across the intestinal epithelial cells, which are IECs, which this pathogen then activates chronically toll-like receptors. And then you get chronic release of inflammatory cytokines, IL-6, IL-8, IL-21, IL-22, which will further upregulate NF-kappa B and stimulate CD4 T cells to differentiate into inflammatory T helper 1, T helper 17, and CD8 cytotoxic cell populations. So this is really important. And I know this is kind of boring, but really, really, really important to discuss the nature of biofilms and how that is integral to whether we have a pro-inflammatory or anti-inflammatory state. Briefly, we have endotoxin here, which is the most inflammatory component of gram-negative bacteria. And when that goes across a leaky gut and gets pieces of endotoxin get into systemic circulation, we have a chronic inflammatory state. This is, again, the component of LPS. And this is, again, showing how we actually have in our gut, how a way to actually, we have intestinal alkaline phosphatase, that one, which dephosphorylates lipopolysaccharide. I don't remember learning about that in medical school or even as undergrad. So I find it super interesting how we know we have ways of trying to protect ourselves from this inflammatory process, but these mechanisms get overwhelmed. So we can see the differentiation between healthy and metabolic disease, and we can see how anti-inflammatory signals are going to be generated via a diverse microbiome with good butyrate production and upregulation of PPAR gamma and gut peptides like the GLP-1s, which again are all the rage for weight loss, and good metabolic state versus in a time of disrupted microbiome, either dysbiotic state or poor microbiome metabolic diversity. We have no or low butyrate production, low propionate or acetate production, and thus we have increased levels of inflammation, increased metabolic endotoxemia, inflammation, and blood glucose dysregulation, or what we call dysglycemia. All of it is connected. So again, metabolic endotoxemia, we talk about wanting to bind up that toxin, and I use a lot of serum bovine immunoglobulin, which binds LPS and helps to neutralize other microbial peptides. And there are new targeted peptides as well. You have to use them orally to specifically treat the epithelial barrier and tight junction proteins. I want to argue that I do not use or try not to use a lot of regular L-glutamine. There's a lot of controversy on whether it gets, some patients do get neuroexcitotoxic symptoms. I use a reduced form of L-glutamine that is complexed to alpha ketoglutamate, which really tends to help patients with their energy in their gut. So remember, we want to use short chain fatty acids if a patient has low levels or they can't tolerate carbs. We can use those. We can use butyrate products to help to increase oral tolerance and help patients to be able to tolerate more foods. It's really important. This is another good, this is Yehuda Schoenfeld's paper talking about cornerstone species, that what do we see in autoimmune disease when we start to see these dysbiotic flora are, what do we see that gets us worried? And what is the science showing us for specific disease states? For instance, we can see a disrupted Firmicutes to Bacteroidetes ratio in things like lupus. And so these are things that we see and we know, and we talk about the use of different treatments like probiotics and even even fecal transplants, although that has been really called into question and been really difficult with COVID. So one thing that I really want to talk about is this paper. You should be aware, and the Gut Zoomer does measure this probiotic, this gut pathobiont, which really does drive autoimmunity. The only thing I just want to say is that if you see Enterococcus gallinareum, you really want to have your spidey sense up to say this is really important and really concerning for a bug that induces autoimmunity. So you're going to want to really take that seriously. And this is a wonderful review, 2024, very current, about talking about dysbiosis as a gateway really to the development of autoimmune disease. And this is very new, but we know, in my world, I've been doing this for a long time, very personally. It's been more than 20 years of studying, and it's kind of like finally we have papers that are saying this is valid. This is really important. And so this is a nice, there's a really nice graphic that I would, have that you should pull and even print for your patients that can show how all of this is related. It did not reproduce well here, so I didn't put it here, but I believe it's figure 1. And these are just tables which show a highlighted specific species that if you see these and a patient has, for instance, rheumatoid arthritis, and they have P. gingivalis, it's a huge, warning flag that could have been a direct cause. So either and Prevotella, so P. coprii, P. gingivalis, really important. So these are pro-inflammatory triggers and molecular mimics. For lupus, again, Enterococcus gallinareum is in there. Strep is in there. And remember, people harbor strep in their guts. And elsewhere, you can get strep from the skin to the mouth to the butt, everywhere, lung, everywhere. And then I just wanted to highlight here, we see MS, and we can see pro-inflammatory if we have high levels of Akkermansia. We have overgrowths of methanobrevir and strep. So again, can be problematic. I want patients and doctors to start to leverage known microbiome relationships. And I want to highlight the role of something called E. coli strain Neisel 1917. It is the oldest probiotic on the market. What do you mean you haven't heard of it? It is known as Mutaflor or Mutaflor because it has been used as continuously since 1917 in Europe. It is not sold here in the United States because it is E. coli, and the FDA says E. coli is bad. And I will say, are you a microbiologist? Not all E. coli is bad. You can get it from other countries. And it is a mainstay treatment of choice to maintain maintain remission of ulcerative colitis and other conditions of the gut. So this is Dr. Alfred Neisel, and he was a microbiologist in Germany in the World War I time, and he was trying to find ways to help. As you may know, Germany didn't do so well in World War I. They got blamed for the war. And people were devastated when soldiers came back in Neisel's town. There was a young man who did great, and he said, "How did you survive trench warfare and gas warfare and pandemic flu and all of these things?" And he said, "I want to study you." So of course he did what any microbiologist would do, and he found a bacillus in the young man's stool that actually was able to resist pathogenic bacteria, including those that cause dysentery, like Salmonella. And so this was Dr. Neisel's paper on E. coli antagonism, and he identified the concept of competitive inhibition. So bugs that could inhibit other bugs, and this is the Deutsche Medizinische Wochenschrift. And so this is what it's doing. We see that E. coli strain Neisel 1917 has a relationship between the intestinal microbiome and the host. We know that there's direct antagonistic activity with the inhibition of growth or the killing of pathogenic bacteria and yeast. And we actually change that intestinal colonization resistance so we can get more good guys. We can increase good biofilms and mucin production, sealing tight junctions, immune modulating. anti-inflammatory effects, and the inhibition of invasion of gut epithelial cells by really bad pathogens like Shigella and Yersinia and Listeria and yeast and Salmonella and different forms of other pathogenic E. coli's. So I use a lot of gut and wheat zoomers. It's not a perfect test. There is no one perfect test, but we can screen for root causes of their imbalances, and we can use and leverage these relationships. And I want to show that. I do other breath and antigen or antibody testing if it's necessary, like Candida testing or H. pylori testing. No test is perfect. I often use advanced hormone testing. We rule out mycotoxins and other environmental toxic burdens. And I just will ask, do not test food sensitivities first. If someone has a trash gut, they're going to have an exceptional burden of sensitivities, and you're going to bum them out. And it's going to be really difficult to treat. And remember, we don't want to restrict first. So this was a first case, and I always say Roux-en-Y gastric bypass is a perfect way to ruin your health. And this, in fact, they are not actually doing a classic Roux-en-Y gastric bypass anymore unless someone really needs it for their life, because what they are doing is they are bypassing a portion of the gut, and that area, that limb that remains bypassed is going to become overgrown with bacteria and yeast, and you have to continuously decolonize these patients. It's a fact. And so this is a patient, a gastric bypass patient who on their wheat zoomer is not only going to have the leaky gut, but is also making antibodies against lipopolysaccharide. So you are essentially having an autoimmune response against endotoxin. Wow. So that's really challenging. So we really want to bind that up. We really want to talk to them about all the things because they're in a chronic systemic inflammatory state. We can see low gut diversity, and we see those ratios of Firmicutes to Bacteroidetes that are off, which is likely, sadly, a reason for them to have been prone to obesity in the first place. We can see a ton of problems here. We see that they're not digesting veggies. We see that there's blood in the stool. We see gliadin peptides. We see zonulin. So anti-gliadin antibodies, excuse me. We see zonulin in there. The gut lining is being degraded. We see fats there too. So there's a lot of malabsorption going on. And there's a lot of big macromolecules that are going across the gut lining, causing a lot of inflammation and autoimmunity. We can see very high levels of, not very high levels, but we see higher levels of gram-negative bacteria, Enterobacteriaceae, that's that family. And this is a core relationship I want you to see. The levels of gram-negative bacteria, and remember, those are the LPS producers, including E. coli's, and then lower levels of Akkermansia feeding the butyrate production and slime layer and microbial diversity and metabolic fitness. All of that is part of the disease process. We can see not only that, we have lower levels of good probiotic species, and I'm highlighting Eubacterium and L. reuteri. L. reuteri is really important for metabolic fitness and for weight loss. We now show that. And lean body mass. Eubacterium is associated with longevity. And so we see these relationships. So when I look at this, I go, uh-oh, I've got my work cut out after me. And so sure enough, we have very high levels of E. coli's, and we have high levels of Methanobrevibacter smithii. And these guys, again, classically, we can't diagnose SIBO this way, but it's suggestive of methane producing small intestinal bacterial overgrowth. Again, it's going to be up to you on whether you're going to corroborate this with a breath test or you're going to preemptively treat. And again, we have to throw the kitchen sink at a patient. So as we can tell, their short chain fatty acids are going to be very, very low. And sure enough, at the very bottom, this is important. We have high levels of potentially pathogenic or dysbiotic E. coli and very, 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 very almost negligible levels of good E. coli strain Neisel. And so you can actually use Mutaflora, and I will use that in the cold months of the year because I have to ship it from over the border and have patients do that as I'm looking at and treating the levels and layers and layers of care. So case 2, this is a patient who has been an infertility patient and had a ton of different procedures, instrumentation, and she had resistant yeast. And this is a huge problem for these patients. We can see the yeast there. We can see a huge Firmicutes to Bacteroides imbalance and pore diversity. And the yeast have really gotten, this is SIFO, small intestinal fungal overgrowth. And so I'm not, for the purposes of this slide deck, I'm not going to go into it, but just talking about that's where you're going to want to start. And you're going to have to open up biofilms, and you're going to have to do a lot of different things because these patients tend to be really resistant. And not to end with nightmare fuel, but I'm going to. This is case 3, my patient who was ziplining in the Caribbean. And he's a police officer, and sure enough, he had several things going on, but he had a combination of swelling in his legs, and he'd had a history of corneal ulcer. That was not why I was testing him, but he had a lot inability to gain muscle, inability to gain weight, and a lot of symptoms of floating stools, and a lot of symptoms of malabsorption, specifically fat malabsorption. And sure enough, we see that he has non-celiac gluten sensitivity, among other things. And sure enough, his pancreatic elastase is very low, and he has fat malabsorption. And this is a very comprehensive process of replacement that's needed. Many different aspects, and it's going to be trial and error for the patient. So this is a wheat zoomer pearl. So there is an association between non-celiac gluten sensitivity and low pancreatic elastase. So this is very similar. We see that in celiac disease, and we just started to have recognition in clinical publications about that. And so really it indicates the risk of fat malabsorption. Pancreatic elastase should be above 400. When you see that in the 100s, it's really, really concerning. And so you're going to need to give them digestive enzymes, even pancreatic enzymes and digestive bitters. All different cholagogues are needed, things to help get the symphony of hormones and communication between the organs happening. And so I told you this is a police officer who had a history of a corneal ulcer, and he had lymphedema. And as it turns out, he had been ziplining in the Caribbean, and about months after he came back, he had this really weird, really severe corneal ulcer. And as it turns out, these little parasites, so Mancinella, which is a worm, it's a helminth, actually can go into the eye, and it can actually destroy lymphatics. And so we saw that in here, and we were able to diagnose and treat the patient accordingly. So again, it's all of these layers. Again, he's going to have to treat the parasite. He's going to have to treat the overgrowths. We can see these relationships here. We see he's got indicative of bacterial overgrowths, methane producers, strep, etc. So again, I always talk about it like Shrek. Ogres are like onions, which have onions have layers, and ogres have layers, and the microbiome is always layers. So it's lots of overgrowths. So we say go big to go small, go big and then go small. If there's yeast, treat the yeast first, then treat the bacteria, then treat the viruses. Do not go after Lyme first. Always open up detox pathways. The liver is paramount, and you can't treat if you can't poop. So remember, SIBO, small intestinal bacterial overgrowth, is not a bacterial problem, at least not alone. It's a motility problem as well. We have the layer of the nervous system at the cellular level called the migrating motor complex, and we really want to support that. Remember, we want to stimulate the vagus nerve to upregulate the parasympathetic nervous system, and we talk a lot about wanting to improve autonomic nervous system coherence. So again, treat those layers. I really do personalized treatment according to testing. I look at using layered and pulse dosing of herbal and prescription antimicrobials if I have to. Sun fiber is something that I love. It's been shown to work really well in concert with Xifaxan. Remember, methanogens are really tough. It's layer after layer of treatment. Sometimes we have to use other off-label products because methanogens are ancient bacteria. They're archaea bacteria that are most like eukaryotes, not like regular bacteria. Neomycin is actually toxic, so I use a lot of neem instead. And remember, SIBO will beget SIFO, and you have to treat both. We'll often look at advanced breath testing to also rule out hydrogen sulfide producers and pulse dosing of advanced biofilm busters. So again, these are other things that I'll use. We want to use prokinetics, supplements, prescriptions. I use a lot of LDN. I really highly suggest that you look at the research on LDN to downregulate toll-like receptors, downregulate chronic inflammation, support autoimmunity. For inflammation, I use a lot of SPMs. Tough cases are going to be, drastic. And sometimes I'll even use things like elemental diet. We want to use vagal toning, brain retraining, neurofeedback, anything that I can get my hands on for the patient. It's really complicated. So remember, great moments are born from great opportunities, said Herb Brooks, a miracle advice coach. We've got great opportunities to help our patients. And so I thank you for your time. I'm here to answer questions. So thank you very much.
Great. Thank you, Dr. Z. That was amazing. I loved all the studies. Can't wait to dive into those and all the clinical pearls. I've got a couple of questions that came up during your presentation.
Perfect.
First one from Lindsay. Is there any relationship between secretory IgA and IgA nephropathy, as she often sees low SIgA in her IgA nephropathy patients, but there's some concern. But some concern is there if we work to increase it in the gut, will it increase in the kidneys as well and contribute to kidney damage?
We don't have evidence that if you use enteric, meaning taking, say, bovine SBI or things like that, that you are going to cause kidney damage. Now, you really, we don't have any studies on that. You really need to follow the patients. You need to be looking at their actual protein levels, both just how much foamy urine are they making and getting protein levels to actually see what their nephropathy is actually doing at the level, plus their labs. We have to look at that. And that's where I would be I wouldn't start with things like that, but it is absolutely true. Their secretory IgA is depleted because they've got end stage disease. And so it's like chicken or egg, but you're seeing the effect. You're seeing that low secretory IgA because they've got this process that's already happened and they have autoimmune disease. So I haven't seen any evidence of the fact that you're going to get more damage doing that way. But obviously, we want to be very judicious for a patient that may not be able to handle a lot of amounts of proteins too. So these are things that we may even use more like peptides or smaller things, smaller molecules, or be more judicious when we're giving that, to try to improve their diversity and thus their own secretory IgA levels. It's a really good question.
Yeah, great points. Thank you for that. Kate wanted to know specifically that form of L-glutamine that you mentioned. I knew this was going to come up, and it did.
Right. Am I allowed to use names?
Sure, in the Q&A, yeah, absolutely.
Yeah. So because patients will be reading, and there's a lot of controversy because you need large amounts of L-glutamine to make a difference. For enterocytes, these enterocytes are really hungry and you need lots of grams. I'm not talking about short-term use of like a lot of the gut healing products that'll have a couple grams in it. We're talking about dozens of grams, 10, 20, much more, that some of my patients are more unable to tolerate. And I use a product by Perk, Perque, called EnduraPack. And it's the only one that I've found. I know Russell Jaffe, who's the founder of Perk, and he's a doctor, a real wonky doctor that I've worked with, and he developed this reduced L-glutamine, which basically one serving size, like a 1.5 grams, is equivalent to like 10 grams of regular L-glutamine. And I find that it really helps with energy levels. In general, it's very well tolerated. So I've had good luck with it.
Super. Thanks for sharing that. Are there any other questions out there? I'm not
I knew that was going to be asked.
Not seeing anything else in the comments, but we've got another minute left of this presentation. So if you've got a question, please submit it in the comments. We'll stick around. All right, here's another one. How do you
Absolutely. So so number one, I love these questions. They're awesome. So anti-thyroglobulin antibodies, that's a real problem. So if you know, maybe you don't know, but talking to the royal we, all of us. So these antibodies are in different places. Thyroglobulin antibodies are more intrinsic to the thyroid gland itself. So this patient's at really, really high risk of actual thyroid gland destruction, more rapid and more symptomatic, especially if they need thyroid hormone. These are patients that I won't give natural desiccated thyroid to. They're more likely to react against it, have problems with it. They're in general going to be more symptomatic, have just more ups and downs, more problems. If their TPO is within the regular range, they're also at risk here of Graves because there is an overlap between thyroglobulin and Hashimoto's Graves. So they might actually cycle between hypo and hyperthyroidism. So you're going to also want to look at TSI, thyroid stimulating immunoglobulin, and thyroid binding autoantibodies. This is a very on my sleeve topic. Most doctors don't know how to treat it. So if I'm teaching, I really want patients, their doctors to know this. And so when we see this, number one, we talk to the patient and say, we need to go into damage mode now. They need to be gluten-free. They need to be knowing that they're at very, very high risk. And that's where I will test them. And yes, if they're living in a moldy house or school. I see this in my school teachers. I just posted this on my personal social media about this. I had a patient who literally brought in a petri dish of what her water damaged temporary building outside, one of those temporary outside shelters that she teaches in. It's terrible, completely mold ridden, and has been so sick, and it caused her autoimmune thyroid disease. I have a point. But yes, it absolutely can be associated with biotoxin illness. It can also be due to endocrine disruptors. It can be due to Lyme. It can be due to, EBV. It can be due to other viruses. So it means having a little bit of a shotgun approach and saying, okay, we really need to dig deep and try to find if there is a known trigger. Sometimes there's not. Sometimes, we can't figure it out, and they can have really high thyroid antibodies. But it means having kind of a come to Jesus talk with the patient about what's going on, that they're at very, very high risk for their thyroid to be destroyed, and how do they want to proceed? Because a lot of these tests are very expensive, and you have to triage that. But doing things like a TGF beta test, instead of a whole, I'll work with a specific lab that is in my office to get a whole SIRS panel. But if they can't do that, they can do that, just to say, am I barking up the right tree? To say, is the immune system being activated by something? And then you may do a mycotoxin test, or, you have to get the family on board at that point to be like, this patient has a really significant thing going on. And, allopathic medicine is just going to say, oh, nothing to do until your thyroid gets destroyed, and then we'll put you on thyroid meds. But these patients tend to do really, really poorly, even on thyroid meds, because they have much more inflammation. Also, please make sure you have a thyroid ultrasound to see if they have a goiter. And like I said, rule out Graves' disease, because you can also get thyroid eye disease with patients with high thyroglobulin antibodies.
Wow, such a thorough answer. Thank you. And I think that is our final question. Oh, another one just showed up. Are you okay to do one more, Dr. Z? Okay. Anita said, I read somewhere that gluten can fuel cancer growth.
Yep. See, so that's where I will not use the regular glutamine, because there are literally, I will not name names. There's like one guy who is the guy on the internet that talks about cancer being fueled by L-glutamine. So I do not use it in the form that the glutamine is getting recycled to glutamate, which glutamate is more cytotoxic and can be used as more fuel. So if you're concerned, again, if there's a family history, I probably, unless it's like they've got BRCA mutations or check mutations where they're really like, I don't want to use anything that can be potentially carcinogenic. Good luck with that, with our environment. But that's where I would be more judicious, and I'd have a discussion. But in general, general, that's one of the reasons why I try not to use a lot of just straight L-glutamine. Just in general, it's not that I don't find it helpful. It's just patients are so sick and you've got a fire hose of things to do. I don't just use L-glutamine, or, it's not a primary treatment that I use a lot of, or I'm going for this other, type, and I'm doing it in a really judicious way.
All right. That brings us to the end of the presentation today. Thank you, everyone, for joining. I hope you gained some clinical pearls to take back to your practice with you. And thank you so much, and have a vibrant rest of your day. Take care, everyone.